Well, after an introduction like that, I'm sort of tempted to stop now, actually, whilst we're ahead. Well, thank you very much for coming out on this wet evening. It's 2 years ago yesterday that I last addressed the public lecture here. And was anybody here at that lecture? Okay, so there's a couple of slides you've seen before. Don't worry about those. The other thing I wanted to do is find out who you all are, actually. With a public lecture, it's quite varied. So, students, who are the students? Yeah. Students at Westminster, all of you? Oh, some from elsewhere. Do we have any physicians here? So just the two of us being insulted by the professor so far. Okay, fine. And any press? Excellent. Okay, so let's move on from there. I trimmed this down from a 3-hour lecture to something around about an hour, and we'll see if we can zip through it. So, from about 200,000 years ago, man spread out from the Rift Valley in Africa and throughout the whole of the world. And soon after, a few tens of thousands of years later, cannabis cropped up in all of the directions in which man moved. So cannabis and man have been together for many, many years, indeed, probably 20,000, 30,000 years. But further than that, the early civilizations started in the big river valleys of China, India, Egypt, Mesopotamia. And as soon as there were writings that were emanating from those areas, cannabis was mentioned in these different words through all the way up to, and Jimmy noted, all the way up to Ireland in 1845. So the cannabis has been used both for its hemp qualities, for rope, for sails, for materials for paper, but also as a medicine. And all of these writings that you see here have referred to the use of cannabis as a medicine. Now, our story with epilepsy starts in Sumeria. It's an area which is now southern Iraq and became Arcadia and Sumeria again. About 2200 BC, these cuneiform runes spoke about something called Hand of Ghost. And Hand of Ghost, it's now agreed, was nocturnal epilepsy. And interesting enough, in the same runes, the treatment for it was Azula in Sumerian or Azulu in Akkadian, and that was cannabis. So if anybody thinks we've done anything new in the last few years, they did it about 3,000, 4,000, 5,000 years ago. Now, the Greeks and the Romans spoke a lot about cannabis, but we don't have any real evidence of their use in epilepsy. So we fast forward to the 12th century to Al-Mayusi, at the peak, really, of Islamic medicine. And this is quite interesting here because he prepared an intranasal administration of cannabis leaf juice to treat epilepsy, so an early example of drug delivery technology. Moving on into the 15th century, al-Badri reported the treatment of the son of a caliph's chamberlain. And here's an interesting point. It cured him completely, but he became an addict who could not for a moment be without the drug. So this gives us a hint of the content of this cannabis here. This cannabis used would have probably had a significant content of THC in it. The modern literature on cannabis was really started in 1839 and subsequent writings in 1841 by Sir William O'Shaughnessy. Now, Sir William O'Shaughnessy was a true polymath. This was a surgeon who was a professor of chemistry at the University of the Medical School of Calcutta. He also researched intravenous therapy, laid 3,200 miles of telegraph around India, was a forensic chemist, designed his own electric motor in the 1840s, was an expert in galvanic electricity, underwater conduction, and the rest. And it's the nature of the polymath which allowed people to think beyond the immediate tunneling or siloing of science. And one of the things you mustn't do is get siloed in science. You'll miss lots of opportunities. But he spoke about, in his presentation then, of infantile convulsions being treated very well with Indian hemp. Now, a few years later, Lay, based on O'Shaughnessy's report, was successful in treating a 9-month-old infant. And by 1848, Christensen seemed to be just reiterating what O'Shaughnessy had said some 10 years earlier. Now, when we look at what Willis said, it would seem from this quote that at that time, cannabis was probably not the first-line treatment in his practice, because he'd used all other treatments before he came to it, and we'd wonder why that might be. McMeans, in 1860, had treated successfully children with cannabis, with cannabis and children with their seizures, and then moved on into adults. And then we come to Sir John Russell Reynolds, often quoted as Queen Victoria's physician. I doubt this. I think he was a physician in the royal household. The Queen Victorian's physician would have been physician in ordinary to Her Majesty. But in his paper on the 22nd of March in the Lancet, 1890, he said, "I found hemp very useful for treating epilepsy." So you would think the use of hemp, with hemp extracts, hemp and cannabis, the same material, for the treatment of epilepsy was really beginning to move on quite well at that time. So around about the same time, at the end of the 19th century, we saw the emergence of a lot of pharmaceutical preparations containing cannabis, and it was quite often the case, as with Dr. J. Collis Brown's Chloridin from about 1895, that there was a tincture that contained opium, cannabis, capsicum, and chloroform. And I have to say to you that if you had chronic and severe pain today, this would probably be a better quality of analgesic than anything else that modern medicine has produced. But you can see on the label here that they were claiming the treatment of epilepsy again. You can still get J. Collis Brown's in Boots, actually, except it doesn't have the opium or cannabis in it anymore, but it does have the capsicum and the chloroform. And capsicum, very important medicine. So, as I said, things are beginning to look favourable in terms of beginning to develop medicines from the cannabis plant for epilepsy. And then in the early 20th century, the vilification of cannabis began. The tinctures were removed from the pharmacopoeias in the UK and the US in 32 and 41. And then the Marijuana Tax Act, under the control of Harry J. Anslinger, in 1937 pretty much put an end to the availability of cannabis and cannabis materials both in the US. This was further reinforced by the United Nations Single Convention Treaty on Narcotics in 1961, which entitled every nation throughout the world to enact their own prohibitions. That was seen in the Misuse of Drugs Act 71 in this country, and of course the Controlled Substances Act in the United States. And this really dampened research into the area of cannabinoids. And the research that was being done in the 80s and early 90s was mainly done by the National Institute of Drug Abuse, and they were actually looking for the modes of action which produced all the side effects and really didn't contribute in terms of medicine. However, it wasn't all doom and gloom. Throughout that period, science was proceeding in the area of cannabinoids. And so in the first half of the 20th century, the identification of cannabinoids in cannabis was underway. And in fact, cannabidiol, and I'm going to talk to you a lot about that this evening, was actually isolated ahead of THC. The major work on isolating and synthesizing THC was done by our very dear colleague, Rafi Meshulam, who I'd say is the father of modern-day cannabinoid science, and certainly deserves a Nobel Prize, I think. Now, that was then followed by a discovery of an endocannabinoid system. A receptor was found. Once you have a receptor, then you have pharmacology, then you have a science, and then you're really back in business. So we found the CB1 receptor, CB2 receptor. This is only in the last 20 or so years. And Rafi said, well, if there's a receptor, there must be a ligand. So he proposed and decided what it might look like, went to find it, and he found it. The first one was anandamide. Anandamide is Sanskrit word for bliss. And the second one is 2-AG, 2-arachidonylglycerol. So by the time we get to founding our company in 1998, there is the basis of all the science you'd need to be able to move forward to create medicines. The problem is the environment really wasn't very friendly. At that time, if you were caught with a pound of cannabis in Oregon, you'd face life without parole. Patients with haemophilia died in jail using cannabis for their secondary effects. Now, you could also track, and this is a very common sort of slide you see these days, Google hits and that sort of thing. You can track the emergence and the explosion of cannabinoid research through the number of papers written. You can see that blip in the 70s and 80s, which is very soon quenched, and I think that's really the Just Say No, 1982, the war on drugs, and there was a very low period. But by the end of the 90s and then into the 2000s, cannabinoid research really took off, and you can see the forming of the company there. Now, in that blip in the 70s and 80s, there was some work done, and in fact, you'll see that there's three controlled trials in one series carried out in very small numbers of patients with very mixed results. The first 2 showing some very promising results, signals that you perhaps wouldn't want to ignore, and the other 2 not finding anything at all. And so this is all we had in terms of any clinical exposure running into the early 2000s. Now also, I'd say this diagram, for anybody who's in cannabinoids, is as famous in cannabinoids as I think Marilyn Monroe by Andy Warhol is in art. This diagram was put together by our friend Angelo Izzo from Italy, and it describes by that time the wealth of pharmacology that was beginning to emerge around the cannabinoids, and it began to explain why in the previous 100, 200, 300 years or a couple thousand years there were so many different uses of the cannabinoids, because you can begin to see nearly all systems in some way or other are affected, and this work has been validated very well. Now, as you know, in this modern day and age, if you don't have the letters OME on the end of your science, you aren't a modern science. So we have an endocannabinoid dome now, and this diagram was put together by Vincenzo Di Marzo, shows the formation and metabolism of anandamide and 2-arachidonyl glycerol, and the targets. Now you can see CB1, CB2 targets there. Those are G protein coupled receptors. Others have been found. GPR 55 was the first, and then the others following on, and we can also show you the TRP receptors here, TRP1, which is the capsicum receptor, and TRPM8, which is the menthol receptor. All targets, and here's some of the effects of the cannabinoid of the plant cannabinoids at these targets. Now, our own team has been able to contribute to this work, and some of them are in the room today, in the areas of cancer, neuroprotection, epilepsy, inflammation, metabolic disease, and my own personal favourite, energy management and bioenergetics. So I think it's probably a reasonable time at this point to introduce you to our company. So by 1997, the government was feeling really quite uncomfortable here and wanted to separate the stigma of cannabis from which was surrounding the use of cannabis in patients with MS, osteoarthritis, and there was the spectre possibly of wheelchair-bound MS patients being pushed up Whitehall in the pursuit of legalisation. And being very pragmatic, the British government said, if there's a medicine, we need to find somebody to develop it. And Lord Williams gave this undertaking in the House of Lords, which basically said, if a product could be developed and approved by the regulators, then that product would be rescheduled from the Schedule 1, the illegal schedule that cannabis was in at that time, to an appropriate schedule. Obviously, at the same time, we had the new research coming through, and it was the case that modern technology now allowed us to prepare extracts again according to modern pharmaceutical standards. The use of whole extracts of plants and plant medicines really fell out of use post-war because of the inconsistencies in their preparation. And the government and we recognise the value of the regulatory process, which provides very, very important protections to patients. So 19 years further on, we've evaluated 14 cannabinoids. We've contributed over 80 peer-reviewed articles, and I've sent cannabinoids to about 35 countries around the world for research. Something over 500 scientists and technicians in our company have now completed 40 phase 2 or 3 studies. We have 55,000 patient years of exposure, of safety exposure on these materials, and our first product, Sativex, has now been approved in 30 different countries around the world for the relief of MS spasticity. And we grow, extract, produce and manufacture all of our medicines in-house. I think it's probably fair to say that GW is the world leader in cannabinoid science today. We've been ably assisted in this endeavour over the last 20 years, 18 years, by about 30 universities around the world, and I just managed quickly to put Westminster in yesterday. I realised, Jimmy, you weren't on there. So I've borrowed your logo from your website. I hope it's the current one. And some, on the right-hand side, are the who's who of cannabinoid scientists. Let me tell you a little bit about Sativex, our first product. This is a standardized extract, a whole plant extract, the first time a plant extract was made as a pharmaceutical in modern times, thought to be the most complex pharmaceutical development in modern times, which contains principally THC and CBD as the cannabinoids, but also minor cannabinoids and non-cannabinoid constituents. As I said, it's been approved in 30 countries for spasticity and multiple sclerosis, but also in Canada for neuropathic pain in multiple sclerosis and in cancer pain. And there was a very strong rationale. Empirically, we prepared an equal quantity of THC and CBD because that's the type of cannabis that was used over the last hundreds and thousands of years. Recreational cannabis that was you found in America and people grew themselves was almost entirely THC. So it's the introduction of CBD which was new in modern times and was really trying to replicate what had been written about in all those publications papers over the previous 100 years. And each of these contribute their own pharmacology and interact together. The CBD takes away some of the intoxication and adverse effects of the THC. So there are over 100 cannabinoids in the cannabis plant, which is the unique source of cannabinoids. You don't find them anywhere else. Cannabis plant is a member of a family which only has 2 members. The other member is hops. So you have hops and hemp are 2 members of the Cannabaceae family. And here's our pipeline here, and you can see at varying stages from early development through to approved products. And there's, for us now, a real focus in orphan pediatric indications. So the perturbations of the developing brain in terms of epilepsy, autism, neonatal hypoxic ischemic encephalopathy, and psychotic illness in children. So let's talk a little bit about the plant and how it produces cannabinoids. The cannabinoids are a primary output of the plant. They're not a secondary output of ripening or something like that. And they are produced through a codominant monogenic control. It's straightforward Mendelian genetics. And our scientists have also been able to map the alleles for the plant to decide whether to produce a cannabinoid or not, which cannabinoid it will produce, and whether the side chains are long or short side chains. And so therefore today we are able to dial into this plant the chemical content that we want from the plant, and that's why we coined the phrase a chemovar. So a chemovar is a variety which is characterized by its chemical content. And at the moment we have over 200 such varieties in our laboratory. So how do we grow them? We take these clones, these cuttings, so that our entire crop is identical. Okay? There's no gene manipulation here, it's just good gardening and straightforward Mendelian genetics. They're put into little plugs, into propagating plugs, left in a polytunnel for about 2 weeks, and a root system forms. They're then potted up and to produce the botanical raw material. That is then cut and dried under very special conditions, all validated and sorted out. And then we make a whole extract of the plant using liquid carbon dioxide under pressure, and it acts as it has the permeation characteristics of a gas and the solvent characteristics of a liquid, and was used in the hops, tea and coffee industry, but had never been used in the pharmaceutical industry prior to us using that. And what you end up is in this pot here, something that's like a cross between treacle and violin bow wax. It's a very sticky, very difficult material with which to start, just the last thing you would use to make a medicine. It's good for sticking tiles to the floor and that sort of thing. Although the Home Office did make me promise when I started this program not to produce white powders from our work. We now have some slightly yellow ones, but not white. So then we analyze them to ensure that we know exactly the content of these materials, formulate into a final product, and that's the botanical drug product. The first step is under good agricultural practice, and interesting enough, 20 years ago, there was European good agricultural practice to cover the growing of botanical medicines, and the next 2 steps under good manufacturing practice, which is absolutely essential for producing any drug worldwide. So here's an example. We take 2 clones, a THC clone and a CBD clone, take them through to the BDS, quantitate them, and then mix them together in equal volumes to give you Sativex. We've also researched an extract, a CBD extract, which has CBD and a lot of the other components. But for Epidiolex, our treatment for epilepsy, we purified it further, and we actually have a yellow crystalline matter there, not white powder. And that's the material that I'm going to talk to you about this evening. Just in case you're wondering whether we can grow enough of it, we have a number of production glass houses. This one is 45 acres, and at any one time we grow 275,000 plants in this glass house, and we got a few of them. So let's go back to the beginning of our evaluations of the cannabinoids in epilepsy. And we do have Professor Ben Wally in the audience today that did most of this work, or certainly his students did at the time. So the first in vitro work was looking at hippocampal slices where they had a 64 electrode array measuring the depolarizations across those slices. And you could induce these seizure-like depolarizations by the introduction of 4-AP or removing magnesium from the environment. And you can see on the left the very active depolarizations, and on the right, with cannabinoid, very settling down. This is actually a take from a video. When you see the video, as Ben says, it's like hamsters making love under a blanket, apparently. That's the scientific bit. We then moved on into our in vivo studies, went through the whole range of in vivo studies that are used throughout the industry to look at anticonvulsant drugs. And just a few examples here. What we were able to see was that the cannabinoids, both CBD and its relative CBDV, were able to reduce seizure severity, decrease seizure duration, and decrease mortality quite markedly. So, and there are anticonvulsants on the market, for example, Keppra, which is one of the world's leading selling drug, which is positive in only audiogenic shock and none of the others. So we had a full house, really, of in vivo outcomes. We also noticed that in the animals that seized, there was an upregulation of certain genes. And you see these gene 1, gene 2, gene 3, gene 4 with different potential functions. And you can see that the addition of CBDV in this case was able to attenuate that upregulation. And when we looked at that attenuation a little bit closer and divided the animals into those that responded and those that didn't respond, the responders had a complete attenuation. It's quite an interesting thought that this might give us a sort of biomarker for what might be going on. Now, also the technicians that were handling these animals noted that those that were treated with the standard anticonvulsant therapies were demonstrating really quite marked side effects, but they weren't seeing that with the cannabinoids. So we undertook a study on what's called static beam, where a mouse is encouraged to walk across a little beam, and you measure it, look at its balance, how far it travels, and how many times its foot slips. And you see in the left-hand panel here, with increasing doses of the classic anti-epileptic drugs, you see that the performance is very poor and dropping off with increasing dose. And on the right-hand side, you can see that neither CBDV nor CBD caused any detectable deficit, unlike those drugs. So a nice laboratory validation of what had been observed. Now, in anticipation of taking these programs into the clinic, we also had to carry out the standard gamut of preclinical work, both in pharmacology and toxicology, and this would be the standard for any pharmaceutical worldwide. So now we had arrived really with two candidates, CBD cannabidiol, the original one, and CBDV, a little bit rarer, and with a slightly different profile, which was interesting enough to us. And we had decided and intended at that time, so we were now about year 2011, 2012, we had decided that we would take CBDV forward into our full development. But then everything changed. And this was the birthplace of Epidiolex. So if we just think about it, we'd been thinking about using the cannabinoids in epilepsy since 1998. We held a continuing medical development in California in 2000, where we further expounded those thoughts. And our work on CBD did not go without notice. Fred Gardner, who edits a journal called O'Shaughnessy's, recognise the name from Sir William O'Shaughnessy. This is the alternative science journal for the cannabis advocacy, and it does make excellent reading, actually. Very, very good observational work. He founded a website called projectcbd.org, and he attributed its founding to his discussions with us. And what he was trying to do was encourage American, mainly Californian growers to stop growing just high THC cannabis, which was the recreational form, and start to grow cannabis containing CBD. It was said in 1998 at an international conference on cannabis by Professor John Morgan that didn't Dr. Guy know that CBD is an inert component of the cannabis plant, and that the entire pharmacology of cannabis can be explained by the pharmacology of THC, to which I replied, didn't the professor know that American cannabis only has THC in it? So CBD, in our mind, has always been the most predominant and most important. Around about the same time as the growers in California were beginning to think about, let's grow some higher CBD-rich cannabis, the work from Ben's laboratory at Reading was beginning to find its way into the published literature. So the challenge was taken up by Harborside, a dispensary in California, and others. And one way or another, some CBD-rich extracts were made, and they were given to children in the 2010-2011 timescale. One was featured, Jaden was featured, his father treated his son who had Dravet syndrome. We'll talk about that in a little while. And it started then to zip round the internet through Facebook, and indeed Paige Figgy read about that, and we'll talk a little bit about Charlotte in a little while. But out of the blue, in August 2012, I received a phone call on behalf of a family in California whose son had a complex form of childhood epilepsy and had about 60 to 70 absence seizures every day, plus some cognitive decline and developmental decline, educational development decline. And they had seen the effects of these vernacular extracts in California, and they worked one day, didn't work other days, some days they made children worse. And they wanted a pharmaceutical formulation and approached us. We knew that under the legislation in the US, you could treat individuals provided their doctor was happy with that. But it would have taken me about 6 to 9 months to get that medicine to the US. So we asked whether they'd bring Sam, and his name's Sam, I can tell you that because his father's a journalist and wrote a big article in Wired about it 2 years ago. We brought Sam to London, and he was treated very successfully by Professor Helen Cross. The deal was they came to London, they had the CBD, but when they went back to America, they couldn't take it with them because it's a Schedule I illegal substance in America. But there was light at the end of the tunnel. We then started to put together a program to be able to send CBD to the US, and the FDA had seen our CBD data because it was part of our Sativex dossier in previous years. Now, very, very unusually, what happened was the news of Sam's treatment and successful treatment just went viral on the internet. Not only are patients and patient advocacies reading this, but also doctors. So we convened an ad hoc meeting in a basement in New York 3 weeks later with top US epileptologists from throughout the US, members of the National Institutes of Health, National Institutes of Neurological Disease, the ex-director of the orphan division of the FDA, and others. And we put together a plan which said we would try and use this route of individual treatments to introduce CBD into these very needy patients. Many doctors had patients or parents banging on their doors looking for these materials. But to do it one at a time was going to be very difficult. I'll show you one on the next slide. Professor Davinsky in New York picked up the phone, rang up the FDA and said, what about it? And they said, well, why don't you do it 25 at a time? So we then started to be able to add to our expanded access program in the United States, patients 25 at a time. But each time we added an IND, an approval from the FDA, we had to go through these 10 steps. Now, they're doable, but take a very, very long time. And that's why batching these into 25 at a time was very, very helpful indeed. By May 2013, Sam was starting to take the drug in the US. However, there was a real firestorm that was around the corner from us. And that was Sanjay Gupta, the chief medical correspondent for CNN, put together a program around young little Charlotte Figgy. Now, Charlotte had Dravet syndrome, and she had been given some high CBD oil and had an almost miraculous decrease in her seizures. And the news of that, again, on CNN, just really rocketed around the US, and there was this tsunami of interest. Following that, 17 states passed laws to enable CBD to be administered, mainly, interestingly enough, to over 18s, didn't really help the children. And of course, as you now know, there are numerous hemp oils available. And later that year, Catherine Jacobson published a survey with Dr. Porter on 19 of the original children that had been treated with the vernacular presentations with some really quite remarkable positive outcomes. And there perhaps may be some confirmation bias there, but again, those signals were interesting to us. Our program continued to grow, and to the extent now that we have 20 physician sites, and 6 of those states that passed laws came to us for our materials, and the FDA have approved treatment of over 1,000 patients. Now, at the last cut of this data, it shows that the frequency of total seizures in the entire patient group was decreasing between 40 and 60%, but around about 40% for most of them. The dark blue bars here were the 40 patients in that group that had Dravet's, one of the worst and catastrophic syndromes, and they were showing really very good results. Now, this is open data, not considered satisfactory to take a product to a regulatory approval, but a very, very strong message, especially with the numbers. That data set was also the subject of a Lancet article in December 2015. So by now, we actually had a product, we'd given it a name, it was called Epidiolex, had a nice box with labels on, and we'd designed a program of pivotal trials to be able to take this product through to the regulators. So let's just think about the patient group that we're thinking about. We thought that these epilepsies were really, really rare, and that we were probably speaking on the phone to the people that were concerned. Actually, there's nearly 500,000 children in the US that have epilepsy, and a lot of these are rare ones. They split into 40 plus different syndromes. But if you look at the pie graph on the right-hand side, this is a very famous chart, quoted very often. When you treat patients with epilepsy, after the first drug, 47% of them will be seizure-free. When you add a second drug in, you add an extra 13%. By the time you add the third drug in, and yes, they do take 3, some of them take half a dozen, and these are quite toxic drugs, you add only another 4%. Past that, you don't get much further. So you have this refractory group of 36%. So now we have about 140,000 American children, and the figures are the same in other parts of the world, who have these rare, catastrophic and severe epilepsy syndromes with no treatment at all. Now I'm going to give you a little bit of let's talk a little bit more about what we see in these children. This epilepsy is catastrophic. They have uncontrolled seizures which cause brain injury. Often you'll see status epilepticus, where they seize and the seizing won't stop until they're admitted to hospital. The sudden unexplained death from epilepsy is 4 to 7 times more prominent than those that are treated. And this syndrome is attended by or accompanied by a series of behavioral problems as well: intellectual development, cognition, language, autism, psychiatry. It's often the case that parents would say to me, "Doctor, I can live with the seizures, but I can't live with the other issues. My 10-year-old with a development age of 4, or no contact at all." So very, very difficult. And the only treatment for these is a polypharmacy cocktail of extremely potent and quite toxic anti-epileptic medicines, anticonvulsant medicines. So here's my little historical aside. Remember that 36% refractory number. So in 1857, not far from here, Sir Charles Locott was boasting that he knew how to treat epilepsy by using potassium bromide because it quelled sexual excitement, and it was thought that hysterical epilepsy in women around about their menstrual cycle was to do with too much sexual excitement, so they'd use some potassium bromide. By the end of that century, bromide was the absolute treatment, in fact until 1912 when phenobarbital came for epilepsy. And Turner reviewed at the end of the century 3 series of patients treated with bromide, and out of 873 of them, 299 remained with no benefit. That is a refractory population of 34%. So in about 120 years, we've gone from 34% refractory to 36% refractory, even though all of these drugs have been launched along the way. So it might indicate that all these drugs might not be touching exactly the right spot, or we may just have a part of this syndrome which is clearly not treatable. So let's see what starts to come out of the formal pivotal program that we mounted for the FDA. In our first study in Dravet syndrome in these children, what we saw was a very highly significant decrease in reduction in seizures. This is a median percent reduction in seizures, both with convulsive seizures, which are the key seizures to look at in this patient group, and indeed in all of their seizures when we looked at them. That data was published about a year later in the New England Journal of Medicine. And then we announced the results of our first Lennox-Gastaut study, another very catastrophic syndrome in children, again quite marked and highly significant decrease and reduction in the median percentage of seizures. both drop seizures and total. In Lennox-Gastaut syndrome, you look for drop seizures where the child is just going to drop to the ground. These are the children you quite often see that wearing the helmets. Okay? And we also did a study where we reduced the dose. Those two previous studies were at 20 milligrams per kilogram per day, and we also went down to 10 milligrams per kilogram per day. And you see that both of those work very well, were significant or highly significant. When we asked the patient or the carer about their global impression of change, in both our LGS1 and the Dravet study, we have a highly significant change in favour of CBD versus placebo when looking at very much improved, or slightly improved. And we see the same situation in the two-dose study. And here you can see that the CBD caregivers were up to 2.6 times more likely to report an improvement in these patients. The safety profile of these cannabinoids compared to the anticonvulsant drugs that these children are taking is extremely benign. And the only real thing that we see in this profile here is a transient elevation of liver transaminases. But this is not accompanied by an increase in bilirubin. It doesn't meet the criteria for liver damage. And all of these resolve even when the patients remained on the drug, or in some cases they came off the drug, but they all resolved. It's not unusual to see that. You see that with valproate, sodium valproate, one of the most common drugs these children take. But this is a very mild to moderate profile of side effects compared to some of the side effects with the other medicines. And the regulators require us to do a whole slew of other studies. Here we're looking at pharmacokinetics, we're looking at renal impairment, hepatic impairment, thorough QT studies looking at electrophysiology of the heart, human abuse liability studies. Do people like it as much as THC containing cannabis, for example, and drug-drug interaction studies. Now, many of you may well have read about or heard about the drug-drug interactions that have arisen with Epidiolex, and this is why we have to treat these medicines as sensible and proper medicines. What we find is that in the presence of clobazam, another popular drug for the treatment of Lennox-Gastaut, not approved in Dravet, what we find is we get a larger improvement. Cannabidiol on its own is providing very worthwhile improvement for these patients, but in these patients it's due to an elevation, possibly an elevation of a metabolite, N-desmethyl clobazam, we get an improvement over and above. We see that as an advantage. Others see that as a drug-drug interaction, and I think the patients and the clinicians see that as an advantage. So following on from those initial studies in Dravet's Lennox-Gastaut, we've now moved on into a tuberous sclerosis complex and infantile spasm. Tuberous sclerosis are children born with benign tumours in their brain, kidney, heart. These tumours resolve, but 70% of them are left with seizures, and about 40% autism spectrum. There's cognitive impairment and neurobehavioural disorders as well throughout these children. So these children exhibited all those other issues that the anticonvulsants don't really treat and sometimes make worse. And infantile spasm program will be taking us into very young children, 6 months of age. At present, we're down to 2 years. So on the far right-hand side of this spreadsheet here, you can see that we have started and commenced our application to the FDA for approval in Lennox-Gastaut and Dravet. That's a rolling application which started in July and will be completing in the next few weeks. Now, a lot of you will know that a formal pharmaceutical development takes between 5 and 10 years, sometimes up to 15 years, and that the clinical part of that would normally take 5, 6, possibly 7 years. What is interesting to see is that in this Epidiolex program, from the first day of the formal adoption of the program in Q2 14 to filing the dossier was just 3 years. And it's the expanded access program that helped us greatly to be able to speed up that program. Now, you might say, well, it's rather quick. Have you skimped on the data? No, the data package is very comprehensive with over 1,500 patients in the safety profile. Orphan drugs generally have a much, much smaller group of patients and patient safety data. We have over 400 patients in our dossier that have been on the drug for over a year. So where does this take us in the future? Home straight, everybody, don't worry. So the future of cannabinoid research. I call it research of perturbations in the developing brain, because I think if you wanted to know what the cannabinoids do essentially, I think they make, guide, create, develop, refine the brain. They then repair and replace and regenerate. And that is, I think, their fundamental role. And if you go back through evolution and evolutionary biology, cannabinoids have been around for 700 million years. And I think we've had a significant amount of co-adaptation with these materials, especially in the development of the brain. So remember this picture here. We've tidied it up a little bit. Sorry about the contrast on your projectors here. Us doctors usually have slightly sharper projectors, but I think you'll see most of these things. What we have here is the basis of lots of mechanisms of action, and as you probably know, that pharmacology and modern-day pharmaceuticals all are looking for mechanisms of action. But when you've got a polymodal medicines from nature, and this is very often the case, aspirin being one of the earlier cases, many mechanisms of action, then it's really almost like looking through a reference library to decide what you must know. So we tend to concentrate more on systems. What are the systems that we need to be thinking about that are involved in the phenotype, the pathophenotype we're looking at? And in epilepsy, we're thinking about inflammation, anticonvulsive effects, neuroprotection, of course, we need cell survival and cell cycle, neural function, and plasticity. And it's plasticity I'm going to talk to you a little bit about. All of these systems, by the way, need very good supply of energy, and therefore mitochondrial function is absolutely essential in maintaining homeostasis in these systems. So let's go back to thinking about cognition behavior. So remember the parents saying, I can live with the seizures, but it's all those other things. Well, these are the things, the developmental delays, the language. Some of these syndromes, the children lose language entirely. Their movement disorders, their social interactions are poor. One mother saying, I've smiled at my daughter for 2 years, she's never smiled back. The autism spectrum disorders, depression, self-harm, quite an issue in these groups of patients. And also, patients that would have a classic diagnosis, say, of autism will also experience seizures. And the anti-epileptic drugs that have been used to date, it's pretty well recognized now, they contribute to the worsening of these issues. So do you have total seizure control, but these issues worsening, or where is the balance? Can we achieve both? So we began in our expanded access program, and the clinicians that were doing that program began to start looking at the children and looking at these extra responses. We had a narrative very early in the program where the child's school report from their special school came back saying language has exploded. The school had no idea what had happened. The child had about 3 words and was talking in sentences. We see that time and time again. And in this group of 27 subjects, using a quality of life assessment developed by Sabaz in 2000, in this group you see quite clearly an improvement in their cognition scores and their cognition domains here, also the sociability. And that, in these numbers and these graphs, is actually what the parents and the patients are seeing. In another group of 18 patients with tuberous sclerosis, again from our expanded access program. Remember, this is data that the regulators would not normally accept, but really guides us in our development. We've got very good response in terms of their seizure control, but here again, they saw cognitive gains in 85% of these children and behavioural improvements in 67%. And the clinicians said that these improvements were not linked to seizure severity or the relief of seizures. They were unlinked. So back in 2011, Francis Jensen and their group started to think about epilepsy as we do, as a spectrum disorder, not just seizures. In pharmaceutical terms, it's seizures because the only drugs available are anticonvulsants. It's like if the only tool you have is a hammer, then everything else is a nail. And so we wanted to see how these syndromes and these phenotypes can match together. And they saw a connection between the epileptogenesis, the development of epilepsy, the seizures and the excitotoxicity of those seizures, and the cognitive deficits and the autism. And they thought that the common factor here all the way through was synaptic plasticity or neuronal plasticity. And so here's another ohm that's come along. We now have the connectome, the way in which the brain connects together. And this was a very important paper, very much ignored by most clinicians at the time. But even earlier than that, in 2000, Nelson and colleagues began to describe the neuroplasticity that was occurring in the new brain. I don't suppose many of you know that on the day of birth, the brain prunes 50% of all of its synapses. Quite a busy day on that day. So any abnormality of energy management, any other abnormality in that child on that day is going to have profound effect on neuronal function. But as you can see, the senses, vision and hearing, are beginning to develop in the first few days and months. Then language, that important new trick that humans have learned in the last 250,000 years, that's coming out in the early months. And then the higher cognitive functions. Well, of course, when the perturbation or the insult occurs to the brain, dependent where they are on these graphs, will begin to determine what the presentation of the phenotype looks like. And that's why there are so many of these different rare disorders, because they're just cutting slices through this data all the way through. So work has now commenced, and publications actually in the literature for nearly a decade now, showing that the endocannabinoid system is very, very closely involved in neuroplasticity, and that's probably a topic for another lecture and something we might talk about in 2 years' time. But what we do know is that about CBD is it substantially increases neurogenesis, the turnover of new nerves. And it increases in hippocampal neuroprogenitor cells. It increases the proliferation of those cells. Now, having more nerves and more cells may not be right. There has to be the right amount. There are some of these rare conditions of overexpression of plasticity as well as underexpression of plasticity. It has to be right. It has to be homeostatic. Now, if you compare that to the fact that also the anticonvulsants, the existing anticonvulsants, are now being seen to be damaging to neuroplasticity. And to that, I would add large and excessive amounts of THC. In young children, large excessive amounts of THC, in my mind, will be damaging to neuroplasticity, and we have a major program of work looking at neuroplasticity with our cannabinoids. We'll be able to tell you about that a little bit later. So when we think about seizures, intellectual development, cognitive function, these behavioral problems, psychiatric disorders, becoming more common, you heard on the news yesterday. I think we're going to see a tsunami of psychiatric disorders in post-adolescence, both in the US and in this country, in sleep. Then the polypharmacology that the cannabinoids provide, not only singularly, but actually when you put them together, they work rather better. We see a lot of synergies. So we use isobolographic estimates all the time in our work. We will be able to deal with the encephalopathies, those that are created by the excitotoxicity, the actual seizures themselves, but the background neuroinflammatory processes that are going on. And of course, each time a seizure occurs, the brain's strategy is to drop its own oxygen tension to try and stop the seizure, and that causes hypoxia. So you get a post-hypoxic ischemia after the seizure, all of which are really not good events. They all cause marked inflammation. And on the right-hand side of this slide, you see energy management. To us, bioenergetics is everything. If the cell and the system does not have the right quality of energy, it will not be able to maintain the appropriate homeostasis. More on that, and that's probably what Jim was talking about. We believe the answer there lies in quantum biology, and that is a very different lecture. So neuroprotection and neuroplasticity are going to be really key words to look for as we move forwards. So when we come back to some of these rare disorders, you see the ones in which we've done clinical trials are in red there. And we could add in the sorts of conditions, not necessarily the ones we're doing, the sorts of conditions which are characterized by these awful behavioral disorders, the CDKL5, Angelman's syndrome, for example, and tuberous sclerosis complex. So in conclusion, epilepsy can be considered a spectrum of seizures to autism. That's new to in most people's minds. We think it's pretty obvious, but to most people it's new. Cannabinoids in drug-resistant epilepsy are effective anticonvulsants. We've shown that in randomized controlled trials, and improves several aspects of cognitive neurobehavioral function. These improvements are independent of the seizure control. It's not just because you take the seizures away that those improve. People with very good seizure control will still have these cognitive problems. And they may be through matched or overlapping pathways, but distinct from seizures. We believe that a greater understanding of the role of neuroplasticity on the pathophenotype and the therapeutic interventions we might make are going to hold the greatest promise for the future from cannabinoids. Thank you very much indeed for listening. Thank you.